HESI A2 Biology Punnett Square Practice. Question 1. Fungal growth is measured on identical substrates under controlled humidity. Two heterozygous organisms in a mushroom growth trial are crossed for flower pigment, with B dominant to B. Which probability describes an offspring with the recessive phenotype? Option A 0%, Option B 25%, Option C 50%, Option D 75%. The correct answer is Option B, 25%. Each parent can contribute either allele with equal probability. Combining the gametes produces one homozygous recessive outcome among four equally likely genotype combinations. Only that genotype expresses the recessive phenotype under complete dominance. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 2. Fungal growth is measured on identical substrates under controlled humidity. Which observation from a mushroom growth trial is the best example of an adaptation rather than a short-term acclimation? Option A. An animal learns a route during its lifetime. Option B. A heritable feature has become common because it improved reproduction across generations. Option C. An individual temporarily breathes faster at high altitude. Option D. A plant wilts during one hot afternoon. The correct answer is Option B. A heritable feature has become common because it improved reproduction across generations. An evolutionary adaptation is an inherited population-level feature shaped over generations. Temporary physiological adjustments and learned behaviors can help an individual respond, but they are not inherited genetic changes. The distinction depends on heritability and generational change. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 3. Fungal growth is measured on identical substrates under controlled humidity. A diploid organism studied in a mushroom growth trial has 10 chromosomes in a somatic cell. How many chromosomes should a normal gamete contain? Option A 5, Option B 10, Option C 20, Option D 4. The correct answer is Option A 5. Meiosis reduces chromosome number by half, so fertilization can restore the diploid condition. Each gamete receives one member of every homologous pair. The calculation uses the haploid number rather than the number of chromatids present during replication. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Stressed about your HESI A2 exam? We can take HESI A2 exam for you remotely and undetected. We always guarantee a pass and payments only after results. To send us an email, scan the code or tap the link in the description and let's get it done. Question 4. Fungal growth is measured on identical substrates under controlled humidity. One diploid cell from a mushroom growth trial completes meiosis without error. Which outcome is expected? Option A. 2 identical diploid cells. Option B. 1 diploid cell with twice as many chromosomes. Option C. For genetically identical diploid cells. Option D. For haploid cells with genetic differences. The correct answer is Option D. For haploid cells with genetic differences. 2 successive nuclear divisions follow one round of DNA replication. Homologous chromosomes separate first, and sister chromatids separate in the second division. Crossing over and independent assortment contribute to variation among the resulting haploid cells. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 5. Fungal growth is measured on identical substrates under controlled humidity. Cells in a mushroom growth trial join small subunits into a larger biological polymer while releasing water. Which reaction type is occurring? Option A. Hydrolysis. Option B. Neutralization only. Option C. Dehydration synthesis. Option D. Ionization. The correct answer is Option C. Dehydration synthesis. Building a polymer commonly removes a hydrogen from one subunit and a hydroxyl group from another. The removed parts form water while a new covalent bond links the subunits. The reverse reaction uses water to split the bond. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 6. Fungal growth is measured on identical substrates under controlled humidity. Consider the food chain algae, zooplankton, small fish, heron in a mushroom growth trial. Which organism occupies the primary consumer level? Option A. Zooplankton. Option B. Algae. Option C. Heron. Option D. Small fish. The correct answer is Option A. Zooplankton. Primary consumers obtain energy by feeding directly on producers. The next consumer eats the primary consumer, and higher levels continue the transfer. Identifying the feeding relationship is more useful than relying only on an organism's size. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 7. Fungal growth is measured on identical substrates under controlled humidity. A cell in a mushroom growth trial contains 11% non-penetrating solute and is placed in a solution containing 14% non-penetrating solute. Which outcome is most likely? Option A, solute leaves by osmosis until the cell bursts. Option B, water leaves the cell, causing it to shrink. Option C, no net movement occurs because water cannot cross membranes. Option D, water enters the cell, causing it to swell. The correct answer is option B, water leaves the cell, causing it to shrink. Water moves toward the side with the greater total solute concentration when the membrane is permeable to water but not the solute. The external solution therefore draws water out of the cell. Loss of water reduces cell volume without requiring ADP. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 8. Fungal growth is measured on identical substrates under controlled humidity. During a mushroom growth trial, cells synthesize DNA. Which smaller unit is repeatedly joined to build this molecule? Option A, triglyceride. Option B, amino acid. Option C, monosaccharide. Option D, nucleotide. The correct answer is option D, nucleotide. Hereditary polymers are chains of units containing a sugar, phosphate, and nitrogenous base. The order of the bases stores information. Other monomers build proteins or carbohydrates rather than genetic material. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Are you looking for HESI A2 exam actual questions? Visit our website pokerexams.com to access HESI A2 exam study guide with actual questions and answers. Scan the QR code or click the link in the description to get HESI A2 exam answer key today. Question 9. Fungal growth is measured on identical substrates under controlled humidity. A membrane in a mushroom growth trial separates 73 freely permeable particles on one side from 27 on the other. Before equilibrium, which direction describes the net particle movement? Option A, from the lower minus concentration side toward the higher minus concentration side using ADP. Option B, from the higher minus concentration side toward the lower minus concentration side. Option C, equally in one direction until all particles stop moving. Option D, only toward the side containing larger molecules. The correct answer is option B, from the higher minus concentration side toward the lower minus concentration side. Random molecular motion produces a net flux down a concentration gradient. Movement continues in both directions, but more particles leave the crowded side than return to it. At equilibrium, motion continues even though net transfer becomes zero. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 10. Fungal growth is measured on identical substrates under controlled humidity. Organisms in a mushroom growth trial need a rapidly available source of chemical energy. Which class of biological molecule is most directly used for this purpose? Option A, steroid hormones. Option B, mineral salts. Option C, nucleic acids. Option D, carbohydrates. The correct answer is option D, carbohydrates. Sugars can be broken down readily during cellular respiration to release energy for ADP production. They also serve in short minus-term storage and structural roles in some organisms. Genetic polymers and inorganic ions do not serve as the usual immediate fuel reserve. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 11. Fungal growth is measured on identical substrates under controlled humidity. An organism in a mushroom growth trial displays a recessive flower pigment under complete dominance. Which genotype must it have? Option A, BB. Option B, BB. Option C, BBB. Option D, BB. The correct answer is option B, BB. A dominant allele masks the recessive allele in a heterozygote. Expression of the recessive phenotype therefore requires two recessive copies. The observable trait alone cannot distinguish the two dominant minus phenotype genotypes. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 12. Fungal growth is measured on identical substrates under controlled humidity. In a mushroom growth trial, a researcher changes temperature and measures germination percentage. Which factor is the independent variable? Option A, the number written on the data table. Option B, germination percentage. Option C, the final conclusion. Option D, temperature. The correct answer is option D, temperature. The independent variable is deliberately manipulated to test its effect. The measured response is the dependent variable. A clear experiment changes one main factor while keeping other relevant conditions as constant as possible. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 13. Fungal growth is measured on identical substrates under controlled humidity. In a mushroom growth trial, a microscopic cell is observed to contain a circular DNA in a nucleoid region. Which broad cell type best matches this observation? Option A, animal cell. Option B, fungal cell. Option C, prokaryotic cell. Option D, plant cell. The correct answer is option C, prokaryotic cell. Cells without a membrane-enclosed nucleus keep their chromosome in a nucleoid region. This organization is characteristic of bacteria and archaea. Eukaryotic cells instead package most nuclear DNA inside a membrane-bound nucleus. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 14. Fungal growth is measured on identical substrates under controlled humidity. In a mushroom growth trial, allele B is dominant for a flower pigment. Which genotype is homozygous dominant? Option A BB, Option B BB, Option C BB, Option D B. The correct answer is Option C, BB. A homozygous genotype contains two copies of the same allele. When both copies are the dominant form, the dominant phenotype is expressed and no recessive allele is present. A heterozygote contains one of each allele. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 15. Fungal growth is measured on identical substrates under controlled humidity. In a mushroom growth trial, individuals with an inherited trait survive drought more often and produce more offspring. Over many generations, what is the most likely population-level result? Option A. The allele associated with the trait becomes more common. Option B. The environment directly writes the same mutation into all organisms. Option C. Every individual develops the trait by need. Option D. The population immediately becomes a different species. The correct answer is Option A. The allele associated with the trait becomes more common. Heritable variation causes individuals to differ in survival and reproduction under environmental pressure. Those with an advantageous trait contribute more copies of the associated allele to later generations. The change occurs in the population's allele frequencies rather than through purposeful modification by individuals. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 16. Fungal growth is measured on identical substrates under controlled humidity. A dividing cell in a mushroom growth trial shows that chromosomes align at the cell equator. Which mitotic stage is shown? Option A metaphase, Option B prophase, Option C telophase, Option D anaphase. The correct answer is Option A, metaphase. Spindle attachments position duplicated chromosomes along the middle of the cell. This arrangement allows sister chromatids to be pulled toward opposite poles accurately. Chromosome condensation begins earlier, and nuclear re-formation occurs later. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 17. Fungal growth is measured on identical substrates under controlled humidity. A researcher examining a mushroom growth trial identifies a molecule whose main role is storage and transmission of hereditary information. Which class best matches that role? Option A carbohydrates, Option B nucleic acids, Option C mineral ions, Option D lipids. The correct answer is Option B, nucleic acids. Polymers built from nucleotides encode biological information and help direct protein production. Their base sequences can be copied and transmitted between cells or generations. Energy-storage molecules do not carry information in the same sequence-based manner. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 18. Fungal growth is measured on identical substrates under controlled humidity. In cells observed during a mushroom growth trial, the cell checks replicated DNA and prepares division proteins. Which cell-cycle stage is being described? Option A G1 phase, Option B prophase I, Option C G2 phase, Option D cytokinesis. The correct answer is Option C, G2 phase. After chromosome duplication, the cell produces division-related components and verifies that replication is complete. This preparation occurs before mitosis begins. Earlier growth and DNA synthesis stages have already taken place. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Stressed about your HESI A2 exam? We can take HESI A2 exam for you remotely and undetected. We always guarantee a pass and payments only after results. To send us an email, scan the code or tap the link in the description and let's get it done. Question 19. Fungal growth is measured on identical substrates under controlled humidity. During a mushroom growth trial, a contracting muscle cell loses its ability to carry out lipid synthesis and detoxification. Which cellular structure is most directly affected? Option A ribosome, Option B mitochondrion, Option C smooth endoplasmic reticulum, Option D Golgi apparatus. The correct answer is Option C, smooth endoplasmic reticulum. Membrane enzymes build lipids and process certain harmful compounds. The described failure therefore points to the compartment specialized for that work. Other organelles may support the cell, but they do not perform this immediate task. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 20. Fungal growth is measured on identical substrates under controlled humidity. A researcher conducting a mushroom growth trial changes temperature and records germination percentage each day. Which factor is the dependent variable? Option A, the researcher's name. Option B, temperature. Option C, the hypothesis wording. Option D, germination percentage. The correct answer is option D, germination percentage. The dependent variable is the outcome measured in response to an experimental change. Its values are compared across treatment levels or groups. The manipulated factor is the independent variable rather than the measured response. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 21. Fungal growth is measured on identical substrates under controlled humidity. During a mushroom growth trial, one group receives the experimental treatment and another is kept under normal conditions. Why is the normal group included? Option A, it provides a baseline for comparison. Option B, it removes the need for repeated trials. Option C, it increases the treatment dose. Option D, it guarantees the hypothesis is correct. The correct answer is option A, it provides a baseline for comparison. A comparison group shows what happens without the tested treatment under otherwise similar conditions. Differences between groups can then be associated more confidently with the manipulated factor. A control does not eliminate all uncertainty or replace replication. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 22. Fungal growth is measured on identical substrates under controlled humidity. A specimen from a mushroom growth trial has a chitin- containing wall but no chloroplasts. Which cell type is most consistent with those traits? Option A, animal cell. Option B, plant cell. Option C, bacterial cell. Option D, fungal cell. The correct answer is option D, fungal cell. Chitin is a major structural material in the relevant biological feature walls. Plants use cellulose, and animals do not have cell walls. Bacterial wall composition and cellular organization differ from those of fungi. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 23. Fungal growth is measured on identical substrates under controlled humidity. During meiosis in a mushroom growth trial, homologous chromosomes exchange matching DNA segments. Which effect can this event have on offspring? Option A, it converts RNA into DNA in every cell. Option B, it prevents all genetic variation. Option C, it creates new combinations of alleles on chromosomes. Option D, it always doubles chromosome number. The correct answer is option C, it creates new combinations of alleles on chromosomes. Exchange between homologous chromatids produces chromosomes containing maternal and paternal segments in new arrangements. Gametes can therefore carry allele combinations not found together on either original homologue. The process increases variation without normally changing chromosome number. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 24. Fungal growth is measured on identical substrates under controlled humidity. A heterozygous organism from a mushroom growth trial is crossed with a homozygous recessive organism for flower pigment. What fraction of the offspring is expected to show the dominant phenotype? Option A, 1/2. Option B, 0. Option C, 1/4. Option D, 1. The correct answer is option A, 1/2. The heterozygous parent produces dominant and recessive gametes in equal proportions, while the recessive parent contributes only the recessive allele. Half the combinations are heterozygous and express the dominant trait. The remaining combinations are homozygous recessive. The evidence from a mushroom growth trial applies this principle in a concrete biological setting. Question 25. Fungal growth is measured on identical substrates under controlled humidity. Which source creates new heritable variation in a population studied during a mushroom growth trial? Option A, mutation in DNA that enters the reproductive lineage. Option B, use of a trait during an individual's lifetime. Option C, temporary change in body temperature. Option D, identical mitotic copying without error. The correct answer is option A, mutation in DNA that enters the reproductive lineage. A change in genetic sequence can create a new allele when it occurs in cells that contribute to offspring. Recombination rearranges existing alleles, while mutation can introduce a novel version. Temporary physiological changes are not transmitted as DNA sequence changes. The evidence from a mushroom growth trial applies this principle in a concrete biological setting.